So, Namaskar everyone. This is Prakash. I'm a principal scientist at Amrita University and Vice President of Genome India International and a co-founding member since 2004, headed by Professor Chittaranjan Kole, who is our president. Today, we take great honor and pride in having Professor C. Prakash, who is going to give an exciting talk from Tuskegee University from Alabama in the States. But before which, we have revered host, Professor Nagendra Singh, who is going to introduce the guest speaker today. So please allow me to take honor in introducing Dr. N.K. Singh, who is a national professor, B.P. Bal chair, and of course, J.C. Bose fellow at ICAR NIPB in Pusa campus in New Delhi. Professor Nagendra Kumar Singh is a well-known agricultural scientist in the area of plant genomics and molecular biology. He's a distinguished alumnus of Benares Hindu University, who after 10 years of PhD and postdoctoral studies in University of Adelaide and CSIRO Australia returned to India in 1991. He has served CFTR in Mysore for over 3 years and GB Pant University, Pantnagar for 6 years. And over the last 23 years, he has been at NIPB in Delhi. Apart from leading several multifaceted, multi-institutional research projects, Professor Singh has guided more than 50 students towards their PhD and master's degrees. And he has more than 400 research publications and a whopping list of 29,600 citations with a H index of 79. He has been a leading scientist responsible for decoding several plant genomes, including rice, tomato, wheat, pigeon pea, jute, mango in India, and established world-class genomic laboratories at ICAR NIPB. In 2011, he led a team of Indian scientists to decode the AHA genome, world's first pulse genome, and first plant genome sequenced entirely in India. Professor Singh has developed high-density SNP genotyping chips of rice, pigeon pea, and mango, and identified a number of genes and DNA markers for rice, wheat, pigeon pea, jute, and mango breeding in an important conservation effort. Professor Singh has invented new methods for analysis of wheat seed proteins and discovered a new class of wheat seed storage protein named trypsin, responsible for excellent chapati and bread making quality that we eat today. He has collected more than 1,000 different wild rice accessions, and its germplasm is available in Indian germplasm databases. Professor Singh is an elected fellow of major science academies of India, including INSA, NAS, and NASI, and is a recipient of several honors and awards, including the prestigious DBT National Bioscience Award in 2002, highest individual award of ICAR Norman Borlaug Award in 2015, served J.C. Bose National Fellowship in 2022, and National Biotechnology Innovation Award in 2023. What a great honor, sir, in having you today. And the floor is yours in introducing Professor C. Prakash. Thank you so much, sir. Please, yeah.
Thank you very much, Dr. Prakash. It is indeed an honor for me to introduce Dr. Prakash, who has been very well known in international agriculture. And I welcome all the participants who have joined online and those who will be watching later on the YouTube. So Dr. Channapatna Prakash, Dean of the College of Arts and Science at Tuskegee University since 2015, has served on the faculty since 1989 as a professor of genetics. He oversees 9 academic departments spanning 18 programs with a budget portfolio of over $20 million. Dr. Prakash is a principal investigator of an $8 million project to build a cancer genomics center at Tuskegee University, funded by the National Institute of Health. He is also a PI for an AIM Ahead grant from NIH to build a center for excellence on the governance governance and ethics of artificial intelligence, data from the US National Academy of Sciences. He also serves as a co-PI on $5 million US aid grant to provide developmental assistance to Nepal. Dr. Prakash's research expertise is in genetic improvement research and food crops of importance to developing countries. His lab was among the first to create transgenic sweet potato and peanut plants and conduct pioneering genomic studies on the peanut. He co-edited two recent books, Sustainable Agriculture in the Era of OMIC's Revolution, and Principles and Practices of Omics and Genome Editing for Crop Improvement. Dr. Prakash has been indeed a global leader in enhancing societal awareness of crop and food biotechnology issues worldwide for nearly 3 decades. Dr. Prakash was recognized for his outstanding work on agriculture biotechnology outreach with the prestigious 2015 Barlog Cost Communication Award by the Council of Agriculture, Science and Technology, which credited him as arguably done more than anyone else in academia or industry to promote agriculture technologies that can help feed the world's growing population. He was also recognized by the Huffington Post as among the top 30 social influencers in biopharma and biotech. He has an active presence in social media, globally ranked as top influencer on agriculture biotechnology with 92,000 followers, generating millions of impressions per month on Twitter at AG BioWord and Facebook at AG BioWord. He also serves as editor-in-chief of highly respected journal GM Crops and Foods. He has been named one of the top personalities who have made the most significant contribution to biotech by Nature Biotechnology. Who is who in biotech? Some biotech's most remarkable and influential personalities from the past 10 years. One of the 100 top living contributors to biotechnology by the scientist, chosen by peer via polling. Biotech Food Herb Hero by Crop Life International, and inaugural Food Influencer Award by the US Soybean Export Council in November 2020. Dr. Prakash is a leading proponent of science-based agriculture development, especially in the use of molecular techniques, including genetically modified crops and genome editing. He provides technical, societal, and ethical perspectives on the issue through his lectures, writings, and social media. He was instrumental in catalyzing the scientific community in many countries to get involved in public outreach on agri-biotech issues. He is a popular speaker, and his views and writing have been published in numerous newspapers and magazines. He has delivered nearly 1,000 public lectures across 80 countries, including venues such as Aspen Ideas Festival, World Food Prize, UN Forum on Sustainable Development, FAO, US Congress, and World Bank. He was invited 3 times to deliver lectures at the Vatican and was fortunate to have an audience with Pope Francis, explain the benefits of golden rice. He served as a panel manager for the USDA's Biotechnology Risk Assessment Grant Program as a member of the Overseas Advisory Committee for the Department of Biotechnology in India. Chaired the Minority Affairs Committee of the American Society of Plant Biology and served on the USDA Advisory Committee on Agriculture Biotechnology under US AG Secretaries Don Glickman and Ann Veneman. So this is a brief introduction of Dr. Prakash, and he is very well known. He doesn't need actually introduction, but the new people who may not be knowing, we have put together this introduction for him, and we are indeed very fortunate that Dr. Prakash has agreed to deliver this lecture on genome editing, its relevance in the Indian agriculture. And now I hand over to the chairman, Dr. Prakash.
Yeah, thank you so much, sir, for the gracious introduction to Professor C. Prakash. So over to you, Professor C. Prakash. So please share your screen, sir. We're quite honored and ecstatic. Thank you so much, sir.
So wonderful. Thank you so much, Dr. Nagendra Singh. It's indeed a very honor for me to be introduced by a man of your stature. You are a global leader, not just here in India, you're recognized all over the world. And I feel very fortunate to be speaking under the auspices of your chairmanship here. But I would like to extend my deep gratitude to the Genome India International for inviting me to speak, and on a Saturday morning, on a holiday, for Saturday is a holiday here, for you to be assembling in front of your devices to listen to my lecture tells me as to the profound interest you have in the subject and the importance of the subject. And so thank you, Dr. Prakash. Thank you for your help in coordinating this event. And of course, Dr. Chittaranjan Kole, who is a father figure in genomics. I don't know how he does that. I think he just released his 178th book, which is unbelievable. I think he should go down into the Guinness Book of World Records as probably the scientist who's written or edited more books than anybody else. But anyway, I thank you. I would like to begin my presentation. And as you know, I want to speak on genome editing or gene editing and talk about its relevance to Indian agriculture. And I recognize almost all of you are genome scientists who probably know more about genomics more than me. And nevertheless, I would like to illustrate some of the examples as to how the genome editing will be of relevance to India, and then hopefully to promote some discussion on that. And more than genomics, my last many years of my life has been spent in trying to foster the public acceptance of GM crops. So what I would like to do is to to bring some of my own experience in working with policymakers, in working with public, the media, even the NGOs, activists who are opposed to biotechnology, and see how we can learn from some of that experience in moving forward to make sure the genome editing does not meet the same fate as GM, and that it would be widely accepted, because this is really a fascinating technology that has wide relevance, it has simplicity and elegance, and so this is going to be of much relevance in improving the food security and contributing to the welfare and well-being of our people in India. So before moving forward to genome editing, I would like to just remind our esteemed audience that when somebody asks you, why do you need to be editing our food? This is a question a lot of people, not necessarily those who are acquainted with agriculture, not necessarily your students, but someone maybe your neighbor or someone you're sitting next to you in a bus or a plane might ask you, why are scientists messing with our food? This is like Frankensteinian in play, meddling of our food. But so one way is to remind them that the food that we grow today we started messing with it, meddling with it 10,000 years ago. Almost every food that we grow today has been modified slowly through selection. And only the past 100 years we've started modifying that through genetics, and the past 30 years through genetic modification, through transgenics, and probably in the past 10 years through the genome editing technology. And so if we had not edited our food, our banana would have looked like this or this, and our maize would have looked like this, like a pencil. And so you could take any crop and you can show this kind of a before and after as to how radically our food plants and all our crop plants have been transformed. And so the tools that we use to transform our crop plants for thousands of years has been selective breeding, which is simply by our forefathers and foremothers, the farmers, chose the best looking plant amongst what they grew and carried the seed forward. And by doing so, they helped eliminate some of the undesirable effects. And we have also used other more traditional based methods using genetics like hybridization. Once the discovery of the genetics followed 100 years ago, we followed a more scientific basis of plant breeding, and this has been very helpful. The accessible recognition that germplasm is the foundation for plant breeding, conserving the germplasm, and using the germplasm using many traditional quantitative genetic tools has been the major force in plant breeding and still continue to be so, even with the advent of transgenics and genome editing. But we have used some even specific tools like mutation breeding, for instance, which creates random mutations, and we pick the best looking phenotype and carry it forward. Okay, there is some sound coming from one of the audience, so can that be muted, please? So,
participants, please mute yourself, all of you except Dr. Prakash.
Yeah, we have done that, please. Yeah, please go ahead. I'm sorry.
Okay, and so in the last 30 years, we've had transgenic breeding, which is essentially introduction of single or a few genes from outside the genome, the foreign genes, and that is what we also call as GMOs. And then more recently, we have started using genome editing, which I'm going to be talking about today. But nevertheless, while we understand the controversy about GM crops, we should understand that plant breeding has always been somewhat controversial. People have been worried about interspecific hybrids. If you look into the literature of 18th century, 19th century, they have cartoons in the newspapers mocking plants for hybridizing or even grafting. And I remember when first hybrid seeds were introduced in India, I mean in Bangalore in the early 70s by Indo-American hybrid seeds, that while farmers loved it and waited even for 2 days to go buy those seeds, there were newspaper editorials asking, saying that why should farmers pay for the seeds? So that's the kind of thinking. And so food, not just seeds, a lot of food innovation has not been readily accepted. It takes a certain amount of time, and as you can imagine, pasteurization, which was invented in the middle of 19th century, took about 100 years before it could be accepted. And so in this context, you need to understand the backlash about GM crops is when GM crops were first approved for use in 1995, 1996. It's been almost 30 years, but we still see considerable apprehension about genetically modified crops, not based on science, because we don't have any scientific evidence that there have been that they are harmful. But nevertheless, many countries have still outright bans, and India, while it allowed the GM cotton, the BT cotton, about 20 years ago, has not approved any new trades since 2006, and primarily because of some of these very orchestrated campaign against this technology. And one thing to remember when we're talking about gene editing is simply a type of mutagenesis. It's a more very specific targeted mutagenesis, but it's not new because we've been using mutagenesis for nearly 100 years using chemicals and irradiation. A lot of useful varieties have been developed. In fact, a lot of our soji wheat that we use in India that has basis in mutagenesis. It was many of those varieties, their parental stocks were developed using radiation. In fact, so much so more than 3,000 crop varieties from mutagenesis have been listed in IAEA. There are probably many more of that, especially when you look at floriculture, a lot of beautiful patterns that you see in flowers, the variegations. Many of them are introduced by mutagenesis, conventional mutagenesis using chemicals and irradiation. And so this is what the GM crop looks like. This is a BT cotton field from Australia. And this is from this one single field, and it is not different fields. And you can see it's like day and night. If you do not spray in cotton against cotton bollworm, your cotton field would look like what it's on the right. But if you have one gene from Bacillus thuringiensis, the cotton field would look like what's on the left. And we see similar contrasting images in corn and many other crops where BT is grown. And so because of some of the controversies and use of foreign genes, many people see, at least from a public perception point of view and from a regulatory point of view, gene editing or genome editing offers better prospects because compared to mutagenesis or traditional breeding, the changes when we do gene editing is very small and it's very precise because you're targeting one gene, probably amongst 30, 40,000 genes that you would find in a plant. And it's very similar to natural changes. And it is knowledge-based because you know exactly what you're changing and you are aware of any toxic or allergenic effects from those changes. And especially if the STN and STO2 methods of gene editing is followed, you do not necessarily introduce any foreign gene. And nevertheless, even just like GMOs, gene-edited crops are required to be tested substantially for food safety and for their environmental impacts. But what is important to recognize is that because of the advent of genomics and our knowledge of many of the critical genes in crop plants, followed by a revolution in so many other omics and proteomics and metabolomics and phenomics, the gene editing provides a very attractive tool to improve crop plants like we have done never before. And so, but nevertheless, we need to look at it as one of the latest tools among the long line of tools that we have used to improve crops and livestock. And so the omics that is be driving much of the plant breeding now is going to be genomics, and that's powered by the next generation sequencing that has brought genome sequencing to be very, very affordable and very inexpensive. And of course, the tremendous increase in knowledge also driven by many parallel developments in computing, especially in the big data analytics and artificial intelligence, and for our ability to track useful traits is fueling a surge in proteomics and transcriptomics, metabolomics, phenomics, and so on and so forth. And so this is contributing to molecular breeding, primarily in the use of markers, which may not necessarily involve transgenics or gene editing, because that way you do not have to get any regulatory approval. You simply use molecular goalposts to track the useful traits and then incorporate them into your breeding program. But if you do not have certain traits, for instance, in cotton, you can screen all the 30, 40,000 varieties of cotton. You will not find a single cotton variety that is resistant to bollworm, for instance. Or you could screen all the 130,000 varieties of rice. You will not find a single variety of rice that is having beta carotene in its endosperm. And so on. For that, you have to bring the trait from a foreign species and then incorporate it into your crop plants. But on the other hand, if there are certain genes that are already present that could be altered, then you could use gene editing. And so Plant breeding now has become a very, very sophisticated operation compared to when I graduated with my degree in plant breeding from University of Agricultural Sciences in Bangalore. Today, you look at the amount of tools the plant breeding, the team that developed in crop improvement have at their disposal. You have genomics, fueled by tools such as DNA sequencing, genetic profile, genetic mapping, the transcriptomics that I talked about with RNA sequencing, expression profiling. So you can look at all that, the tremendous amount of knowledge-based approach to breeding that we have today, identification of candidate genes. And again, bioinformatics is providing a tremendous help here in the genomic selection, QTL mapping, and using of the GWAS methods to see the degree of divergence amongst the candidates and to the walk to your gene. And once the genes are identified, their proteins and metabolites characterized, again, you have we can either go through the transgenic approach or you can go through the genomic approaches. And again, genomic, there are several tools that are available for genome editing, but one of the most popular ones today and the versatile ones is CRISPR-Cas, primarily because of this tool is used in basic research, in biomedical research. So there is literally an explosion of information and improvements of this tool on a daily basis, and all of that accessible to plant scientists for improvement in crop improvement. So I just wanted to show you just a few examples of what could be done through gene editing, and some of it is done by your own scientists here in India, including Dr. Nagendra Singh and his group. So this is one dramatic example where they use at Sainsbury Laboratory to a genome edited tomato for powdery mildew resistant. You can see the control on the top with clear infestation by powdery mildew, a very nasty disease in tomato and many other crops. And through simple gene editing of one gene, you can see the whole tomato plant is free of the disease. And again, just another example for disease resistance is in banana, for wilt disease resistance, where wilt is a very big problem in banana, and you can see it can kill the whole banana crop, and through gene editing, it has been able to provide a control resistance. Again, talking further about gene editing for resistance, this is a citrus where you can see on the control where there is a very clear progression of a disease about 12 days after inoculation. Within some of the gene-edited lines, you can see, especially this line, that it is almost 100% resistance. When you look at the canker is not able to progress and is completely localized. And that is really the incredible power of very precise technology here. Just one more example of bacterial speck disease resistance. You can see how what a mess that bacterial speck causes on the tomato plants, and how beautifully disease-free the gene-edited plant is. And I will share all my slides here through, I will have the organizers send in a way so you will be able to download all this, and you will see the references I have provided, and for every slide I have used from the literature. And this is again another one, a non-browning tomato, non-browning banana. As you know, 30–40% of our banana around the world gets thrown away because it ripens very fast. So when you bring in an element of slow ripening, then you can conserve more banana, and it can even conserve its nutrient, because from literature I've seen, eating a banana like this has a lot more nutrients, less sugar, less glycemic index than this banana. Of course, this is only made very good for making banana bread, but not for probably direct consumption. So this is again genome editing rice for blast disease. Blast is one of the most important plants wherever rice is grown, along with bacterial blight. and scientists have been able to achieve by editing 3 genes for making blast resistance. And this is another one I told you about, the bacterial blight. The major problem, I mean, wherever rice is grown, especially in India, in South India, and bacterial blight is a very huge problem wherever paddy is grown. And this is moving forward, a result from Indian Institute of Rice Research, from our own scientists. I know Dr. Kutub is in the audience here. I'm really honored to have you, Kutub. And Samba Masuri, one of the most prominent varieties of rice, was gene edited to provide increased yield in the rice research station here in Hyderabad, just to show that you could even influence seemingly polygenic traits through perhaps through editing of transcription controls. You can influence even the quantitative genes, such as those controlling the yield traits. So it's not just the qualitative traits like disease resistance or pest resistance that could be altered, but even others too. So moving forward, talking about quantitative trait, there's also by editing, scientists were able to show that they can increase the wheat size and again by editing the number of genes. Editing 2 genes will give you from this into this. And by editing 4 genes, you can see they almost double the size of the grain in wheat. And by editing 6 genes, they were able to make wheat almost probably as big as corn. And so the point is that this provides a tremendous opportunity to progressively increase certain quantitative traits by editing a cumulative number of genes in the plant. And many of you remember that the Green Revolution happened because of the alteration of one single gene in wheat, the Norin-10 gene that was discovered in Japan and introduced into the United States in Washington State. And while Dr. Norman Borlaug was visiting Washington State, identified the incredible potential of dwarf architecture in wheat. Wheat plants were earlier grown for probably about 2 meters long, and it was brought to about 0.5 meter long by introducing a dwarf gene. Now, we can similarly bring about dwarf architecture simply by genome editing, altering the genes related to the stature, and this could be done, and this has been shown in many crops, although I'm showing the example of tomato here, or a wild tomato perhaps. So this again provides us with an opportunity that many crops. I remember as growing up in Bangalore, cotton used to be almost like a wine, and a lot of our crops used to be very tall. And slowly, in the last 30 years, I've seen the stature of a lot of crops have brought shorter because they are more efficient, because they have a greater photosynthetic capability in terms of the plant architecture, and you can pack more plants per acre. In that way, you increase the overall productivity. So there are many ways. Even now, just in the last couple of years in the United States, corn plants have been made dwarf. That has been brought together by breeding, not through genetic modification or gene editing. But nevertheless, you can see after wheat and rice also went dwarf, thanks to Dr. Gurdev Kush, in the discovery of that dwarf gene in the Taiwan collection. Anyway, moving forward, we also have a wonderful research that has shown that one could even increase the nutrient uptake and nutrient use efficiency of crop plants. When you recognize that almost 80% of the fertilizer that we apply to plants today gets lost, they are not taken up. And in fact, in India, the major problem caused by agriculture is really the large use of fertilizer, and it's fueled by fertilizer subsidies and some of the political factors, and even irresponsible use on the part of certain farmers to use more fertilizer just because it gives more yield. But nevertheless, the negative impact of use of fertilizers on the environment has been tremendous. It also causes global warming. Nitrous oxide gas that is released is 300 times more potent than carbon, or 100 times more potent than methane. And so we really need to cut down fertilizer use, and to also to reduce the release of nitrous oxide into the environment. And again, by genome editing, there are many examples where they have been able to improve the nutrient efficiency. And I already showed the slowing down ripening in banana. This is another where you can see in tomato, because we all know in India, in tomato, farmers produce a lot of tomatoes sometimes, and they don't get good yield, and we have seen them throwing on the streets. It's fairly common around Bangalore. And sometimes tomatoes, they cannot even get 1 or 2 rupees farm gate value, and they just throw it away because it's really sad. But on the other hand, if you through genome editing, if you extend the shelf life of tomato from 10 days without refrigeration to 30 days without refrigeration, you really help the farmers, you help the consumer, and more than anything, you reduce the wastage, because tomato has probably one of the highest wastage of any fruit or vegetable here in India and elsewhere. And moving forward, the exciting possibilities also a result from India. Here, it was mentioned the research in NABI, where a banana, they were able to develop golden banana, just like golden rice, by using genome editing. Similarly, even golden rice was been developed without having to put a foreign gene. And that was one of the biggest controversy in golden rice, was because they put a gene from maize. And it's been delayed for almost 25 years. And only one country has just begun growing that. But hopefully, if we bring it through genome editing with native varieties, there might be a better acceptance. They also raised lycopene content in tomato using gene editing. And lycopene has been clearly shown to help improve men in reducing the incidence of prostate cancer. And this is again just one example of health benefits that we could achieve by genome editing. Another health benefit example is how we can develop low glycemic index of rice. You know, for us in India, especially in South India and eastern part of India, rice is the king of our food. And much of our problem in health also come from eating too much rice. And if we can make rice diabetes friendly to low glycemic index, then it is going to be of much better nutritional value. And another recent example that impressed me was how they were able to reduce browning in potato, just like I saw in banana. This is what happens in 48 hours of cutting the potato. And we all know what happened cutting a potato, the phenolics and the browning. And here you can see how that browning has been reduced. And this can also reduce a lot of, eliminate or reduce wastage and improve the food value. And just a couple more examples, who doesn't like a better beer? You know, and trying to reduce the sprouting of barley. Australian scientists were able to use genome editing to make better beer. And finally, I just want to end my talk with how the genome editing is going to be of tremendous value in the using for the climate change. We know in India the effects of climate change. I'm sitting in Bangalore for the first time in December. They have not had any cold days, you know, and our summers are very hot all around India, and we have seen flooding, a lot of salinization issues, erratic rainfall. We know in Madras was flooded a month ago. Even Dubai was flooded about a month ago. And so all of that used to be global climate change, and one of the worst sectors that are going to be affected by this climate change is agriculture. And so we who are agriculture scientists, especially breeders, have a tremendous responsibility and also opportunity to use a variety of techniques and with almost 100 years of our knowledge of drought tolerance, and use very modern tools like genomics, transcriptome, proteomics together to develop plants that are drought tolerant, develop plants that are heat tolerant, and develop plants that can probably cope with submergence. And just a couple of other examples to show how genome editing, this is the wild type grown in high salt water and genome edited plants. You can see the length of roots longer, and they're able to cope much better. Literally hundreds and hundreds of papers published in this area, and very fascinating, and a lot of them many useful. In fact, there's one company in Israel, I think, along with the US, I think it's called Alora. They are trying to genome editing paddy to literally be grown on the seawater. And this is going to be, if it ever happens, this is going to be a tremendous breakthrough, very transformative potential. But nevertheless, a lot of small achievements can be made, and cumulatively they might help. For instance, genome editing to improve the root system in Arabidopsis. You can see as to almost 3 or 4 times better root system, so these kind of plants and crops can thrive in very low water conditions when there is drought. And also for high temperature tolerance. So temperature is a huge factor, especially during flowering and critical stages of plant growth. Tomato plants exposed to 42 degrees centigrade, a temperature that is not uncommon in many parts of India during summer, will kill the tomato plant. But look at the gene-edited plant, 2 strains, especially this one. They're doing really well. And flooding tolerance, also, although this is not due to gene editing, but I just wanted to show this one example where how they took a variety in Bangladesh that can tolerate a rice variety, tolerate flooding, and through very clever genomics marker-aided selection, they were able to transfer this into many crop varieties. But they are very similar. It took almost 7-8 years of backcross to achieve this. But what I wanted to tell you was that we can perhaps do this through genome editing much more quickly. And just one other final, I know there is a great emphasis on millets in India, rightfully. Our Prime Minister talks about it, with the G20 leaders were fed with millets. But many of the millets very hardy and many nutritious, low glycemic index, but they're not necessarily very productive. And genome editing provides a unique opportunity to take the wonderful germplasm, even the wild germplasm that we have in millets and tubers, a lot of marginalized crops in India, and then improve their yield dramatically, very quickly, using gene editing. And the one good dramatic example is they took wild tomato and gene edited many of the traits and make it fully domestic. See, this is 10,000 years of evolution brought into 3 years in the plant. So this sums up a summary slide done by a friend of mine, Dr. Shakil Ahmed, to show how gene editing has implications in breeding. And this is again to remind you in an earlier slide that I showed you how we bring about knowledge in many fast evolving science, including a lot of new technologies in artificial intelligence, sensors that are helping phenomics, even robots and drones. I was just in Bangalore Tech Summit, and I saw some of the very fascinating new developments here in India that is helping agriculture. And so all of them converge into moving forward. And the gene editing also has tremendous other applications that I would not have go into detail, but many of them are very familiar with how this gene editing, or at least the CRISPR-Cas system, has been used in basic research, biomedical research, and also in agriculture research in many ways, to add genes, to knock out genes, to alter the regulatory. To wrap up, I just wanted to say that I have talked about many examples of how genome editing can help Indian farming to improve its productivity, improve the nutrient levels of our food, climate resilience, disease and pest resistance. And India has one of the very our food safety levels are very low, and our food quality levels are very low. It's a very large country, 1.5 billion people, small farms, and again, to improve the quality and safety, gene editing provides one of the tools. Shelf life of fruits, I've already given some examples, but I didn't go into detail some of the quality factors that could be done, like improving oil quality. India last year imported $13 billion worth of vegetable oil, because of our love for samosas and pakoras. We are paying a huge bill. And many of those vegetable oils could be grown here if we simply improve the productivity of our mustard, improve the productivity of sunflower, groundnut, and many other oilseed crops here. And finally, gene-edited crops, unlike GMOs, many countries are lowering the bar on regulation. It takes about 10, 15 years for a GM crop to be approved. It takes about $100 to $150 million of expense just to meet the regulatory burden on GM crops. But on the other hand, India has rightfully lowered the bar and said if you don't have a foreign gene and using genome editing, and then with very minimal regulation, you should be able to approve the crops. But I think the clincher right now, from what I hear, is the Corteva, which has a patent on gene editing. CRISPR is demanding very large sums of money. And I was talking to Ganesh Kishore last night, and he said, look, this whole CRISPR-Cas patent would expire in about 5 years. And so nobody's going to be nobody will be able to charge any patent royalties. And so all our universities and seed companies will have a free hand. And secondly, this technology is developing so rapidly, there may probably many other options than use the patented technology. And so, unlike GM crops, my friends, we have a wonderful opportunity to learn from our past mistakes and see how we can bring genome editing tools very quickly to help the Indian farmers and the Indian consumers. And so one of the most important things we need to do is to communicate the benefits of this technology at every opportunity possible to your policymakers, to your media, to your neighbors, to your school children, in a way that they are better informed and not to be misinformed by many of the big activists that are very present in India. Make sure that we provide evidence on their safety, address any specific concerns they may have about allergy or toxins, and if there are any ethical concerns about modifying life, we need to address that and say that this is something that we are not doing for the first time. It has been done all about. But what is important is we need to be transparent about any risks and also be very, very clear about benefits and work collaboratively with the industry, with the science communicators and journalists in a way that we bring about an improved understanding. And so with that, I just want to end my talk here. And again, thank you very much, Dr. Nagendra Singh, for graciously sharing this and for introducing me. And I'd be very happy to take any questions. Thank you very much.
Thank you, Dr. Prakash, for excellent presentation here today. And you have shown through several examples, simple examples, how genome editing is very, very important for Indian agriculture, and it has many, many applications. Of course, the acceptance of the GM crops and, of course, genome edited crops has been a problem in India due to several groups, pressure groups opposing it, and also partly due to not clear policy from the government also, but recently that has changed. Now government has come into more clarity about the GM crops. So GM mustard was also allowed to be field tested. And also the good news is that for genome editing, the government has made the SDN1 and SDN2 free from regulation, as long as we can prove that it doesn't have any foreign DNA. So actually they don't have SDN1 and SDN2. So that is a very good news. And also DBT and ICR both have allocated funds now for doing genome editing. So things are looking very, very positive. And many scientists have now started projects working on genome editing, and you have shown some examples also. So it is really great, and your presentation was so simple and so clear that people who will be even watching on YouTube later on, they will really appreciate that how important this technology is. Of course, IPR will be over in 5 years, and people can innovate also, alternative way of doing the genome editing. I think we should think about that as a scientist. So really, really appreciate, sir, your presentation here today. And our National Academy of Agriculture Sciences and DBT has played really tremendous role in convincing the government about genome editing. And SOP has been made public. It's very clear that your Institute Biosafety Committee itself can clear it if it doesn't have the foreign DNA. And what you showed that the SDN2, which can, the allele replacement is allowed there. So within the SDN2. So what we take long time for back crossing, etc. Through genome editing, one can do allele replacement within the primary gene pool, and that will be considered as SDN2 because there is no foreign DNA is coming. You will be transferring it anyway by crossing. So through genome editing, you can replace the allele and avoid the back crossing and selfing for several generations. So now there are a few questions in the chat box here.
Do you want me to read out, sir?
Yes, Dr. Prakash, if you can take them one by one, and Dr. Prakash will.
Yeah,
Yes.
please, yeah. Thank you so much, Professor Prakash, for a scintillating talk. Thank you so much for enlightening us. So there's a question from Dr. Saha, Triptapritim Saha. What is the genome editing food accuracy level to consume the naturally harvesting food?
Okay, yes, thank you very much for the questions. I'm going to, can you repeat that? Because I'm also trying to look for that question in the chat box.
Yeah, it's at 10:42. Yeah, it's at 10:42 a.m. Timestamp.
Yeah,
So
got it. From Saha. What is the genome editing food accuracy level to consume than naturally harvested food? Okay, that's a good question. And you know what is the kind of change you are making? And so a good answer to that is the change is so minor that when you take a seed, harvest it and replant it again, the number of changes that happen just naturally is about 1,000-fold more than what we are trying to do with gene editing. You know, there's some very good papers that have done a lot of good genomics work just to monitor the amount of changes, not only through genomics, but also through proteomics and many others. And so the change that you see is very, very specific, very minor, and even put it in the context, the kind of change you're making is just altering 1 grain of sand in a whole beach. And you wouldn't even notice unless you have some very, very sophisticated tools of genomics. And contrast to that with traditional breeding, when you cross one variety of tomato with another variety, and oftentimes I remember when I was working on helping a tomato breeding here in Bangalore, they were crossing with the wild species to bring nematode resistance. So when you cross wild species, you bring literally hundreds and hundreds of unknown genes. Some of them may even cause allergic reactions to you, but many of them we never bothered. You see, even when you do irradiation, it alters literally thousands of nucleotides, gross chromosomal changes. And compared to that, genome editing is a very minor change. It's kind of, think of our conventional breeding as, pickaxe, and our genome modification or even GM as using scalpels.
Yeah. Thank you so much. I hope that answers your question, Dr. Saha. So there's another question from Abhishek Mazumdar. He asked, can genome editing be employed in glycophytic crop roots, mostly displaying negative halotropism or escape from salt into a gravitropic orientation of root systems, or even tolerance to salinity in saline soil for better water and nutrient acquisition and optimum yield in farmers' field, especially in coastal and inland salinity conditions?
That's an excellent question, really. Very thoughtful, because Dr. Majmudar, we need to be very careful working with a wild species, because these wild species, over millions of years, it is, just imagine if you're trying to do something with Parthenium or water hyacinth, two of the most nastiest weeds that you see across India, along with many others. And so many of the crops, like sweet potato, I worked on sweet potato for 30 years. It's practically a weed. It grows so well everywhere. It's weed-like. And in fact, in some countries, it is a weed. And so to take a crop that is highly adapted, glycophotic, or plant that can grow in salinity conditions and drought. Many of those are wild species that are weeds. And when we bring them into agriculture, we need to be careful. That's all I'm saying. I mean, I can go at length as to what all the traits that would make them hope. And so I would rather work with wild species of crops rather than weeds, and bring them into cultivation, just for the sake, because of the chances of those, even with gene editing or genome whatever, chances of them becoming a nuisance feral weed species is going to be much higher.
Yeah, thank you so much. I hope that answers your question, Dr. Mazumdar. Yeah, the next is from Dr. Longcher. He says, could you give a rough information on the current standing of GAC on GMOs release in India?
Well, I'm sure there are many others here who are better knowledgeable on that. From my understanding is GAC now today is scientifically more robust and more facilitating than kind of GAC committee we had. But nevertheless, it's still restrictive in my opinion, because GAC is controlled by the Ministry of Environment. And you look at many of the GAC members, they are not knowledgeable in biotechnology. Some of them are not even scientists. So they're just put there for political reasons. And from what, just look at what they have approved. They've really approved just one, in the last many years, it is the mustard that has been approved, and that is also caught in the Supreme Court today. And compared to that, the way the RCGM is constituted is a much better. There are more scientists in that, and because these are scientific questions, the kind of questions that you're asking now is what is the likelihood of a weed that is gene edited is going to be even more weedier, and what is the likelihood of a rice that is altered is cause allergy, or it's going to cause environmental problem. So even other agriculture scientists who are not experts in food safety or environmental aspects of this can answer this question. So we need to have really professional regulators who can ask, and this should be the outcome of this should be based only on science and not based on some vague socio-economic factors, because we can never answer any of those unless we bring them into the field. And if something is not popular, then it will automatically disappear. Look at today. I remember about 22 years ago when I was going across India trying to help people understand BT cotton. A lot of questions were asked. BT cotton is going to contaminate our cotton. BT cotton is going to destroy farmers. And BT cotton is going to become weed-like. None of that has come true. Today, BT cotton has changed the lives of 8 million cotton farmers, and it produces one of the best protein sources. India has the largest number of milk. India is the largest producer of milk today, and many people may not recognize it. Would not have happened because almost 70% of that milk comes from feeding cotton meal. the highest cotton source that we have today in Indian livestock industry. And so we have 300 companies that are licensed the BT content technology from Mexico. And so we need to be open-minded as we move forward and trust our judgment based on science and not on emotions.
Yeah, wonderful, sir. Wonderfully inserted. Thank you so much. I hope that answers your question, Dr. Longchar. There are several appreciations, but interim, I missed Namita's question. She asked, what are SDN1 and 2 editing methods? Could you detail, if at all? That's, I know it's...
Right. Yeah. Simply put, this is how when we are trying to bring about changes in the gene editing by substitution of the bases, the ST1 and ST1-2 simply involves the nucleotide substitution without bringing any foreign genes in them. And that is the simple way of explaining them. STN3 is where we can bring about those nucleotide substitution by adding foreign genes into them. So this is to again explain that genome editing also can be a vehicle for for genetic modification and for bringing foreign genes into them.
Yeah, thank you, sir. I hope that answers your question, Namita. Moving further, Basav Prabhu Patil, he asks a question. I think this is certainly answered by you. This was certainly answered by you. With the advent of genome editing, should we now give up the transgenic technologies, particularly with reference to funding scenario? What is the future of transgenic crops already in the field? Yeah.
No, that's a very good question. A lot of people ask me, so now that we have genome editing, why should we abandon GM? I don't think so, because genome editing can only edit what's in there. It cannot bring some really novel traits. And so for that, we still need genetic modification. For instance, cotton, I don't think I've seen any evidence so far that through genome editing, you can bring about resistance to the cotton bollworm. Today the BT and probably some vegetative proteins that are found in BT are still the best. And until and unless that day comes when we could probably get that, so we still have to rely on GM. And second, like I said, like for instance, some very novel applications. Did you know that today there is a company that is making really good COVID vaccines in plants by introducing the COVID vaccine? Even the mRNA of COVID can be produced in the plant, and Ebola vaccine is being produced in the plants. And so all that can only be done through GM technology. I would think there is going to be a niche application of GM because of its very high burden that we have for regulatory affairs. It is going to be less and less used, but it will not go away.
Yeah, wonderful, sir. Thank you so much, sir. I hope you can take three or four more questions, if
Sure,
that is okay
no
with
problem.
you. Yeah. So, Dr. Ganpati Mukri, who is also Joint Secretary of GII and a Senior Scientist at IARI, he asks, could you please give glimpses on editing crops for herbicide tolerance consequences?
I'm sorry, I need to look up to see what.
Yeah. Could you please give glimpses on genome editing crops for herbicide tolerance?
Yes. So give me some glimpse on herbicide tolerance. Consequence. Okay, so the consequence of herbicide tolerance, consequence of any crop or any trait is not dependent on how you produce that crop. You see, you can produce herbicide resistance. For instance, recently in IARI released a very good herbicide resistant rice by Dr. A.K. Singh that was developed by conventional breeding. You can do a mutation. The ALS mutants have been produced very well through conventional breeding. Through GM, you can do it. I was last night, as I said, I was talking with Dr. Ganesh Kishore, who developed the herbicide resistance that is used all over the world today, a graduate of Indian Institute of Science. And I'll try to, if you go on YouTube, you can watch his lecture, Dr. Ganesh Kishore, which was just put yesterday, Bangalore. So the question is, what is the consequence of that? Consequence is going to be the same, so it is immaterial how it's produced. herbicide resistance has a tremendous future despite the optics, the negative perception about use of herbicides, because removal of weeds is one of the largest operations done by any humans on the earth. Compared to any other activity, removing weeds is the single most largest effort put by human beings, especially in a country like India. Almost 70–80% of our labor is involved in removing weeds. And just to give an example, all of the paddy that we grow in water, the main reason of water is simply an herbicide. Water keeps the weeds away, and if you can just grow rice, herbicide-tolerant rice that IRI has developed, you can do away with water. The Tamil Nadu and Karnataka don't have to fight anymore regarding Kaveri. And all the problems of irrigated agriculture, salinity, everything comes from the excessive use of water. And so if we adapt herbicide-resistant technology in a very meaningful manner, then it would help increase efficiency of agriculture. And more than that, it will bring about no-till agriculture. Imagine when you can grow crops without tilling the land. Because tilling the land, although is the icon of agriculture, and the plow used to be the icon of Congress party, so that was so important. Plow is the most destructive tool on earth. You see, so herbicide resistance literally liberates the farmers away from that. But there are also going to be problems like herbicide resistance is going to evolve. And that is something you need to deal with scientifically. It's just like when you introduce antibiotics to control a disease, one day your disease will become resistant to antibiotics. But it doesn't mean that you can't use that. It's like knife to using the sharpening. One day the knife is going to get blunt, but you can sharpen it and reuse it again.
Wonderful. I hope that answers your question, Ganpati. Thank you so much, sir. Sumit Naik asks, what are the key scientific, environmental, and ethical considerations surrounding the cultivation and use of GM mustard, and how it might, its adoption impact agriculture and ecosystems?
Yes, again, that's a very big question. And Dr. Nayak, there are many scientists here. I see Dr. Sundar Raj, I was just with him a couple of days ago. And of course, now again, all of them are much better knowledgeable on that. So, but my cursory understanding of GM mustard is that GM mustard was developed for a very specific trait to help in hybridization to bring about male sterility here. And that was a trait with using an herbicide resistance gene. But on the other hand, there are many other applications of GM mustard. If we allow regulation, we could bring in GM mustard with disease resistance, with pest resistance, and many other traits. So each of the concern depends on what trait it was modified with. But as of today, that was developed by Dr. Deepak Pental, the GM mustard that's approved in India, has very little of any of those environmental concerns that are valid, because the trait that's modified is so localized and the application is used in the breeding condition in some limited capacity. I don't think there is anything to be really worried about this GM mustard right now being approved in India.
Thank you. Thank you, Sumit. I hope that answers your question. Thank you so much, sir. So moving further, we have Dr. H.P. Mahesh. What is the probability that the edited allele will mutate back to the wild type allele?
That depends on this particular gene, its sequence, and its stability. We do know that a lot of alleles are unstable and they may easily revert back. You see, and that is where the breeding comes in. So we're not going to just gene edit a crop and just give it to the farmers. A lot of these companies, the universities that develop this simply incorporates into the breeding program. And in any breeding program, it goes through several years of testing to ensure the stability of those genes. And so if a gene is unstable, clearly we would not move forward with it, and they are not going to be used in the breeding anymore.
Yeah, wonderful. Yeah. I'll probably reserve Sumit Naik's another question later point of time. We have Dr. Dev Kate. When it comes to inter or intra-allele gene interactions, How effective are gene-edited lines during hybrid development or recombination?
Again, it's a very good question. This answer is very similar to what I gave earlier. If a particular gene has a very high preponderance for inter or interallelic recombination, then that's going to alter the phenotype of the particular trait, and it is not going to be very stable. And for that reason, a very thorough breeding program would help eliminate any such unstable alleles from being used in the breeding. It doesn't matter how you altered it, even when you bring in some traits by genetic modification. Sometimes, if it's integrated into highly methylated regions, for instance, there might not be any much expression, or the methylation patterns may change over generations, and so all of that could be screened by a thorough breeding program to eliminate such undesirable features of the alleles that we're going to be using.
Yeah, thank you. I hope that answers your question, Dev Kate. So there is one question from Dr. Vipul. What are your views on epigenetic regulation manipulation through genome editing technology?
That's a very good question, and this is something that is just happening in the last few years. I've been watching the genome editing through alteration of some of the genes through methylation. We can not only bring about changes through the direct genetics, but also through the epigenetic modification. And this is very fascinating, and I believe because so many traits are epigenetically inherited in plants and even in human beings. I know there's a very high, the cancer has a very high governance, regulation through epigenetics. And this is, there are already quite many papers on epigenetic modification through genome editing. And I see a great deal of new research that's going to come, and perhaps even commercialized traits of modification through epigenetic modification of genome editing. Good question.
Yeah, thank you. Just keeping in view of brevity of time, there are a couple more questions. If we could request you and probably Professor Nagendra Singh to kindly wait. Yeah. So I'll take probably if you can kindly take the other question from Dr. Sumit Naik. So he asks, yeah, BT cotton has encountered resistance in India, posing a significant challenge. How can we effectively glean insights from the situation to inform our understanding and address the issue?
No, well, Dr. Nayak, the BT cotton has not encountered any resistance from among the users of BT cotton, that are the 8 million Indian farmers. They love it so much that even though India government has not approved any new trait for the past 15 years, I was just in the Vidarbha region 2 days ago in Maharashtra, and I have personally seen not just the BT cotton, the HT BT cotton being grown, and the BT plus VP cotton, the vegetative proteins that are being introduced in corn, and in many, they're all illegal, by the way. What I'm trying to say is BT cotton has been such a spectacular success. The only resistance we see from these are people who have nothing to do with cotton growing. These are the small groups of environmental activists, people like Vandana Shiva, people like Greenpeace, and like that, who are opposed to BT cotton because they have a vested interest in doing so, and probably driven or funded by organic industry or even a pesticide industry. We don't know. But on the other hand, there is really no resistance to that. The only problem BT cotton has been from a scientific point of view is that because all the cotton trade developers have been more or less kicked out of the country by making laws that are very unfavorable for any investment here. There has not been any new developments in science in BT cotton. And secondly, there has been a very large development of pink bollworm because of the extensive use of BT cotton, and that is being addressed by research with both in the academia and in the industry. And so what I try to take a message from this is, let science, investment, and the industry, let them operate without too much interference from the state, from the government. And then whatever problems, the industry and the universities will come up with a solution. And that is what I believe the BT Cotton has shown, that when you bring in a new product that brings value to the farmers, farmers will increasingly embrace it. We have 300 companies producing BT cotton in India. And we have seen India from being a cotton importing country is now a cotton exporting country. And few problems that are in BT cotton are agronomic problems that will be inherent with any crop, with any new varieties that come up. And the only solution to that is more science, more research, and more accessibility to the new technologies, not closing the doors on it. Thank you very much. Good question.
Hello. Sir, this is Dr. Ganapati. I think Dr. Prakash encountered some problem with the mic. I think we have last one question. Again from Abhishek Majumdar. Unless seeds are prepared by the farmers for convenience in post-harvest processing of seeds, can long-owned promising rice genotypes be genome edited to develop unless phenotype without any compromising in yield?
Yes, I don't have much familiarity with that, Dr. Majmudar, but a very educated guess to your answer would be, I'm sure unless mutants have been there in rice, and in terms of yield, it may or may not compromise, but I do believe it may compromise on some of the other factors because, for instance, pest resistance. If it's make it unless, it might invite more pests. And so the only way we could know that is through growing both of them and testing them in the field over several generations. And that is something that is standard for any character, because one thing we have learned in plant breeding all these years is that there is always going to be some cost. When you increase yield, there is going to be some cost. You may reduce in another trait. When you increase protein, for instance, we have seen that the yield comes down. And so the only way we can achieve any desirable combination of traits is through a lot of testing and a lot more science. Okay. I know there are other questions, but I have my email address. I have put in the chat. Yeah.
Thank you so much, Professor Prakash, and thank you so much, Dr. Ganapati, for taking over, and thank you so much, Professor N.K. Singh, for hosting this and introducing Professor Prakash. Dear all revered audience, Professor Prakash is very kind enough, in giving his presentation, which is already shared in the group chat. This was also shared in the GII WhatsApp group. And the talk would also be put up in the YouTube page of Genome India International. We have already tweeted tagging him in Twitter, and we have also we'll also put the recording in the Facebook page, tagging him as well. But before we join hands together in giving him a kind of a wonderful, thunderous applause, may I kindly request all of you to switch on your camera and for a group photograph, if that is okay. We have several wonderful giants, but we have two big giants in the form of Professor N.K. Singh and Professor C. Prakash. And what more, there is a wonderful photographer in the form of me. I'm a very good photographer, and people say that I take very good photos. So let me just try my hands.
Let's see. Screenshot, yeah.
So all of you, please, I'll just take a quick 30 seconds for everyone to join. Yeah.
So actually, Dr. Vidya Gupta there, it's a big honor. I was with two of our students just 2 days ago, Purushottam and Prashant. And my former postdoc, Dr.
So I
Ramanjani
think
Gowda.
a few more, please. A few more smiling faces to appear, please. Yeah. We'll just wait for 15 seconds. Yeah.
I say shout out to Dr. Gowda, who used to be the department head at the University of Agriculture and Department of Biotechnology, my former postdoc.
Yeah. So we are not able to get everyone in one window, but I'll take two certain windows and we will put this. And thank you so much, everyone, especially from the US, especially Gautam and others. It's almost more than a midnight in Canada and in US, 1:30 a.m. And you have joined, the talk here. So say cheese, please. 3, 2, and 1. Yes, I'll take another one so that you'll not curse me. Yeah. Say cheese. Yeah. Yes. Yes, thank you so much. And on behalf of the President, Professor Chittaranjan Kole, General Secretary, Dr. Milind Parke, and the entire core group of Genome India International, and on behalf of Professor Nagendra Singh, as well, who has kindly inserted himself today with his gracious presence. Thank you so much, sir, Professor C. Prakash, for your wonderful talk. Please unmute your microphone for a quick 1 minute, if everyone could. Otherwise, please have a big thumping round of applause to Professor C. Prakash, please.
Thank you, Dr. Prakash. It
Thank
has
you,
been
thank
really
you.
an honor. It's a wonderful presentation, and it is really an honor for me to host you here. And I'm sure many people had logged into YouTube also, and this presentation will be there for a long time to get this information on genome-edited crops, and look forward to more interactions with you on this subject. Thank you so much.
Absolutely.
And thank you all for joining.
Thank you very much, and namaskar. Bye-bye. Thank you for your time.
Thank you so much. And before we all leave, we request all of you to become the life members of Genome India International. So you can visit genomeindia.intl.org. Please ensure that you become life members. So the life membership fee has been waived off to 3,500, and you all could ensure that you yourself, along with your group members, including the PhD fellows, could join. We have had, the very first meeting, all the core members met after almost 21 years. The last time we all met was in the States, way back in 2004. This time, thanks to our finance chair, Dr. Manit Rana, where he organized a conference in University of Agricultural Sciences in Bangalore, where, we all could meet. So, we request all of you to become life members and support the Genome India International efforts under the aegis of Professor Chitta Kole. Yeah. So thank you so much, everyone. So with this thing, and have a great weekend. Merry Christmas and Happy New Year. Thank you so much.
Thank you, Pras. Thank you so much.
Bye-bye. Bye-bye. Namaskar.